This is a production of Cornell University.
Let's start with placement. If we start thinking about placement, when we start talking about placement, what in fact do we mean? Well, if you start thinking about decomposition, you certainly have to think about the compounds, the nature of the compounds, and their presence. So if decomposition is dependent for a single organism to do the decomposition, you certainly can imagine that depending upon what their nutritional needs are, they might in fact have to have multiple kinds of compounds to do the decomposition. Or you certainly would have to have certain types of organisms in the same location to do the decomposition. Go back to this scenario right here. Oops, where was it? Where is that scenario? Go back to this scenario right here. I have one organism doing part of the decomposition and another organism doing another part of the decomposition. Okay? This organism can't decompose that stuff, and this organism can't decompose this stuff. So I need to have either the right kinds of tissue to be decomposed, or I need to have the right combination of organisms to do the decomposition. Okay? So that's the first one, the interdependence of these compounds. The second one is reactivity of the mineral surfaces. Okay? We just finished off talking about CEC and surface areas. Okay? What happens if a material literally gets occluded? It literally gets surrounded by material, and organisms can't get to that material. Also, if I have a mineral surface that actually attracts an organic molecule and holds on to it, the organism has to be able to get to that organism, that organic matter, to actually do the decomposition. Okay, how about climate? And I'll talk about this, and I'll give a couple examples of this in a few minutes, but how about the climate? How is climate going to affect decomposition? Start talking about climate, what am I talking about? Go ahead. Temperature and water are going to affect the rates of decomposition, and I'll have a couple slides to show that in a moment. Soil acidity. As acidity goes down or up, you're certainly going to have toxic effects. The redox state, aerobic versus anaerobic. The presence of potential degraders in the immediate microenvironment, i.e., where the organic matter is, you actually have to have an organism that can do it. Okay? So if this organic material got put someplace that there isn't a community of organisms to do the decomposition, it's not going to get decomposed. Let's talk about water for a moment. Okay? Water, decay, the decomposition is fastest in the moist soils. But it is also really slow when the soil gets waterlogged. So if we look at the relative decay rate versus water potential, so really dry soils, really wet soils, you have a dramatic increase as you increase the moisture, but then you have a dramatic crash. What's happening? I'm losing my oxygen. Right? So moisture controls it. Okay, how about temperature? Okay, overall temperature, overall soil respiration declines at soil temperature. Right? The C10 rule. Okay, biological activity is going to decrease as it gets colder and colder, colder, and chemical reactions themselves are going to decrease as it gets colder and colder, colder, right? So as this is the relative decay rate versus time, as that material increases, the temperature increases, decomposition rates decrease. The relative decay rate. Okay, it doesn't matter if it's fresh material or old material. And again, remember, we have problems with this term right now. How about the chemical factors? The one I would introduce was pH. Okay? Extreme pHs will inhibit decay. Okay? High salinity will also inhibit decay. Okay? Nutrient deficiencies will also inhibit decay. So these chemical factors that are in here also really inhibit or control that decay. So it really is location, location, location. What is going on at that individual location? Some other examples. Surface placement versus incorporation. Now this is integrating a number of these things, but think about this. If I have material that's sitting at the surface versus integrating it in just below the surface, I'm certainly affecting climate. Okay, I'm going to have the heating and cooling, okay, as well as wetting and drying, versus having a constant climatic condition with intimate contact with that organic material. If that tissue is sitting on top of the surface, there's not a lot of organic contact around it, so it actually limits the amount of organisms that can get to it. Okay? It gets pulled down by a worm or whatever, or by a plow. Suddenly that stuff has more intimate contact with the soil material. Population is much more stable, and as a result, it decomposes or consumes it a lot faster. Okay, does that make sense? Here's another sort of thing to think about. Fire-derived organic matter. I know it's your favorite, Mike. This is biochar. Okay? It turns out that rates of decomposition of biochar in some cases are faster and in some cases are slower than the bulk organic matter. Why? It turns out that physical protection and interactions with soil materials has a huge influence on these decomposition rates. Availability again back to that location. How about root-derived carbon? It turns out predominance of organic matter in soil is root-derived carbon. Well, why? I can come up with a couple kind of interesting scenarios there. Why do you think? Any thoughts? Well, is the root-derived stuff just basically less decomposable? Maybe. I mean, it certainly has different structurals, so there certainly would be that argument. But again, it comes back to location. Where are most of the nutrients associated with in the soil? The surfaces. Okay? Where is most of the fresh material coming in that are not root-derived? The surfaces. So what's going to be decomposed first? The stuff that's at the surface, higher populations. Again, a location type of thing. All right, let's keep going. How about physical discontinuity, disconnection? Where it is. Microorganisms occupy less than 1% of the soil volume. What happens if they're not where the tissue is? Okay, spatial and temporal diverse habitats. In essence, I'm fragmenting the habitats, and these habitats are not necessarily connected to each other. Okay? Certainly you can imagine the temporal issue of oxygen. Okay? We just talked about that one. As organisms start to decompose, the CO2 basically builds up and the oxygen is reduced. Okay, there's a temporal disconnection. Spatial disconnection, I plow something in, or the roots shed at a different location and I don't have the population. At that location, I'm not going to get the decomposition. Okay, how about deep soil carbon? It's another sort of spatial. It turns out that more than half of the global soil carbon stocks are deeper in the soil. They're not at the surfaces. Okay, so if the readily available nitrogen and everything like that is at the surfaces, and it's not down here, I'm going to be limiting decomposition rates. Okay, finally, the soil microorganisms themselves. Have I beaten this too far at this point? Are you guys still getting this? All right, I'll move on. Okay, but the important point is these guys themselves are soil organic matter. They are organic matter. And you can certainly imagine the community dynamics of this population is going to affect where that distribution of organic matter is. Okay? Cool? Questions? Go.
How deep is deep?
How deep is deep? And I was hoping somebody was not going to ask that. Okay? Deep in some sense is basically the second meter, but in some senses is also the second foot. I think what they basically mean, the deep soils, they're basically talking about the component of the soil that is no longer interacted by surface organic matter distributions. Okay, so basically the rooting depth. Okay, but that's the rooting depth of the nutrient roots, not the tap roots. Okay, so, but in general, you're looking at 1 meter versus 1 meter or 1 foot versus 1 foot. Go.
You say placement is really important, but like we usually see the organic matter at like the top layer. Is there still organic matter that's not visible like throughout the whole soil?
Throughout the whole soil, yeah. But as you go deeper and deeper, you tend to have less and less. But again, I mean, you go look at this, but still, as you go deeper and deeper, more than half of it's down below in these deeper layers. I think the real issue here is the population, the distribution of the organisms that are doing the decomposition versus the distribution of the material that they're doing the decomposition of, okay, and where that material is, okay. Now, it could be a temporal spatial break. It could be a longitudinal one where you're literally looking at a space change. Okay. It also could be basically right next to each other, but you don't have the right type of material to do the combined decomposition, or you don't have the right organism to do the combined decomposition, i.e. the woody material moving down through the decomposed woody material and so on. That first slide that I showed, that one slide I showed. Okay. So it's, you know, even though you're at the surface, doesn't necessarily mean that you are going to be decomposed. Okay. And then you have the products of that decomposition as well. The reality, no, I don't have that slide up just yet. Okay. The numbers that we're looking at, when you look at this soil pool, I'll go back to this one. The numbers that you're looking at when you're looking at these sort of decomposition products, we used to think that basically 10 to 30 of this percent of this material, this 100 grams that went in, were basically these humic compounds. That's not actually what we see. When we actually look at the soil, we're seeing percentages smaller than 3. Okay, so that means that this pool that was here, all these other carbohydrates, all these other, you know, plant-derived material and animal-derived material, they're actually in the 30%. And the humic material is down in this pool. Does that make sense? Okay, questions?
This has been a production of Cornell University, on the web at cornell.edu.